Hello and welcome to the first webinar of the season for the Nordic Society of Human Genetics and Precision Medicine. And we hope you all had enjoyable summers and are safe from the COVID. We have a very exciting webinar today and a great panel. Before we start, before I do the introductions, I'll just say a few things about upcoming events. In particular, I'd like to point to our virtual meeting, General Society meeting, which will be the 8th and 9th of November. And we will have 2 days, not quite full days as in person, but you're all used to virtual meetings by now, approximately 5 hours each day. And we will have a separate poster site, and this poster sessions, the posters will be available for about a week before the meeting, and there will be videos of the posters, and then there will be a poster session for an hour on the first day of the meeting. And some of the posters will be offered speaking slots in the main meeting, and there will also be prizes for young investigators for some of the top posters. So we encourage you to submit posters from your group, and we hope to see you there. I also want to point to a satellite workshop that we are organizing together with the Norwegian Institute of Public Health on research response to COVID-19 in the Nordic countries. That will be held the week before, Thursday, the 4th of November. And it will also have presentations, panels, and posters on a somewhat smaller scale. And I think the main thing I'd like to say is that the abstracts are due October 8th for both of the meetings. The registration is free for both of the meetings. There is one great advantage to joining the NSHG-PM, which is that we will make the recordings available after the meeting, but they will be available for members initially, and then there'll be a delay, and then they'll be posted for everyone else. So I encourage you to become a member of the society. We are still young. We have had not yet even our first full in-person meeting, but we do try to do a lot of things virtually and through activities with people and organizations throughout the Nordic region. We've held meetings and symposia. We work with research funders, universities, the political establishment, and Nordic cooperation organizations. And we work hard to try to inform through our webinar series, our social media. I think we have a good presence on Twitter and LinkedIn, and we have also worked on the legal working group has worked on a paper on data transfer outside the EEA, and we'll work on other position papers. Through our website, we post news of science and policy that's primarily happening and of interest in the Nordic region. And more recently, we've begun culling papers from PubMed and posting them, ones that are representative of the members of the society's work. I think, and we post our webinars, and we'll be posting our meeting presentations to our website, our YouTube channel. I think I will stop the introduction there. I will say that just before we start the webinar, that during the webinar, we encourage you to use the Q&A button to leave questions for the speakers. And you can do that during the talks. What I'll do is I will ask for a few questions after Camilla's initial presentation, and then we'll have questions after Alexandra's presentation. And then we will go to a panel discussion, but questions can be sprinkled throughout the panel discussion as well. So without further ado, and I will introduce the panelists later, I'd like to introduce Camilla Stoltenberg, who is the Director General of the Norwegian Institute of Public Health. Camilla?
Thank you very much. I'll show a simple presentation of Moba, the Norwegian Mother, Father and Child Cohort Study, and that will serve partly as an introduction to what Alexandra will say afterwards, which goes much more in depth and in detail on what the opportunities for genetic research are in Moba or based on Moba. So thank you very much for giving me this opportunity. Norway just lifted restrictions, COVID restrictions, Saturday afternoon, and I think this is the very first day since beginning of January last year that we've felt that we're not fully occupied with COVID 24/7. So it feels like a relief, but also I feel a little rusty when it comes to other topics than COVID-19 and how to handle it. So now I'll share my screen. I think perhaps you have to stop sharing yours, Håkon, in order to let me share mine.
No, you should be able to share
okay.
now.
Yeah, I'll do that. Can you see it now, or?
Yes, we can. If you can put that in presentation mode. Yep.
I will.
Super. Thank you.
So I've gone back to a title that I've used many times before, when a whole country is a cohort, to revisit that idea. And the Norwegian Mother, Father and Child Cohort Study and the future of Norway's infrastructure for epidemiological research will be the topic of this presentation. So, coming straight out of COVID-19 handling, I feel that it's easy to forget that history is unfolding around us, not only in regard to the pandemic, but also otherwise, because so many of the resources that we have, that we use under normal circumstances to study health and disease, have now been mobilized and are being used to study COVID-19. And we can also see that in just our lifetimes, we've seen unprecedented leaps in the scientific understanding of human biology and society, and that much of that has been applied during the pandemic. These insights are enabled by new technologies, highly organized modern societies, and the entrepreneurial spirit of researchers. And part of this is a revolution in the kinds of data available to epidemiologists who search for causes of disease and good health. And this is where I've stolen the title from, When an Entire Country is a Cohort. This was an article in Science in March 2000, describing the resources in Denmark that encompass the cohort aspects of Danish society or the Danish country. And it's very similar in Norway to what we see in Denmark, with nationwide mandatory registries as a basis and thicker, more in-depth cohorts with parts of the population that are participants in such cohorts for, to date, generations already. And the aim of analytic epidemiology is to find, to discover the causes of diseases and health. And key features of such analysis is the comparison of different groups of people, and that is best done within a cohort. And over the past decades, a growing number of epidemiological cohorts have enabled researchers to make new discoveries, and I think we've all just seen the beginning of that, in fact. It takes a long time because life has to unfold, at least in one generation, and preferably in several. And in recent years, the value of studying trajectories is also increasingly appreciated, the natural development of disease from early stages. And that is also something that cohorts are suitable for. So historically, one of the most important examples of a high impact cohort study is that of Richard Doll and Bradford Hill, which was published in 1954 in order to convince people that there was a causal association between smoking habits and lung cancer, and also later on with cardiovascular diseases and other diseases and other forms of cancer. They linked data from a simple questionnaire about smoking among doctors to information about causes of death from the Registrar General, a very simple cohort. And this design was part of the reason why they managed to convince the public and the authorities that smoking in fact was a cause. The achievements that have been made based on cohort studies since then are impressive, but sometimes not very visible, because the sources of data are not always duly visible and appreciated. And as I have already said, I still think the most important discoveries based on cohorts lie ahead of us. And Moba is a most promising source for epidemiological analysis in the years to come. So why is that? It's a large sample, close to 300,000 people altogether. It's already a long follow-up. Recruitment started in 1999, and it's easy to link to registries, and there are biobanks involved with samples from all the participants. The setting is a country that is a cohort with national ID numbers, as we do have in all the Nordic countries, and comprehensive national registries, and resources to follow up, also with more in-depth sub-studies. So Moba and other Norwegian cohorts, such as the Hunt study, combine size, scope, and depth. And great analytical depth results from the combination of survey data, biological samples, and the possibility of linking records across generations and families. Since it is embedded in a system of national registries, it gives the scope needed for scientific rigor, or that provides the scope needed for scientific rigor. The key is to assess bias in the data against the standard of the whole population of a country. And the potential in these data is still greatly unappreciated. So Moba's key features is that it is a family cohort with mothers, fathers, and children. And recruitment of pregnant women started in 1999 and ended in 2009 when the last child was born into the cohort. Altogether, there are more than 95,000 mothers, 75,000 fathers, and 113,000 pregnancies, of which many are siblings. There are questionnaires, biomaterials, and registry data linked to the original data from Moba. And there are many exposures and many outcomes that are described and where we have comprehensive information already. And about 166,000 samples have been genotyped and are available for analysis. So GWAS is available in 30,000 trios and EVAS in 3,000 trios. Examples of study designs using Moba are, for example, the case cohort design, which I've used some pretty old examples because those are all examples where I've been involved myself. So we've looked at intake of folate supplements in early pregnancy, which was associated with a 50% reduction in the occurrence of autism spectrum disorders, but intake of fish oil supplements were not associated with autism, which strengthened the finding. And there the case cohort design was used. Another design that has been used and is being used more and more in Moba is the discordant sibling design, where we've, for example, looked at paracetamol, which was used in one pregnancy, but not in another in the same mother. And that showed that paracetamol is associated with increased risk of neurodevelopmental disorders in children. And then third design, which is being used, is the natural experiment. So pneumococcal vaccine was introduced in Norway in 2006 in the middle of the Moba inclusion period. Then we could compare those children who were 2 years old when they received the vaccine before the vaccine was introduced, which meant that they did not receive the vaccine, and those who were 2 years old after 2006. They all received the vaccine. So a decline in early childhood respiratory tract infections was found. And in fact, we also found a decline in pneumococcal disease among the grandparents. That isn't from this paper, but that was found in using the same design. And now in Norway's pandemic response, Moba is making a real difference. Moba offers an infrastructure to address some of the most pressing questions. For example, the true prevalence of infection in the population, long-term health effects of mild COVID-19 infection, effects of vaccines, side effects of vaccines, and adherence to social distancing and other measures. And we're using it for all of those issues, and we'll be using it for many, many more questions as well in the years to come. So this is just a brief overview of the data collection, including the COVID-19 data collections that have been going on in 2020 and 2021. I won't go into detail about this, but it's showing the different stages at which we've collected data through questionnaires from the mothers, from the children, and also from the fathers, and the collection of biological specimens. So how can we unlock the potential of Moba? I think there are currently three main challenges, and one is the need for large-scale genetic analysis and whole genome sequencing in particular. The other one is the need for new infrastructures for data storage in Norway, which is essential for the legitimacy of performing large-scale genetic analysis, but also for storage and use of all the other data linked to each other. We're struggling very hard to get all the permissions we need to do the studies that I think we must do. The third one is the need for a modern regulatory framework that handles the complexity. And sadly, I think that's the most difficult one, both within Norway, but also within the Nordic and European context. Perhaps we can use the COVID-19 pandemic to show how important it is to do something about all these three challenges rapidly. So again, this is just an overview of the genetic analysis that have been done so far, showing that there are methylation analysis, telomere length analysis, metabolomics, some sequencing data, and mitochondrial DNA that has been initiated or is already ongoing. And you see to the left, the phase 1 contributors, the different studies, the ERC harvest study, and all the other studies that contributed to the Moba genetics originally, which has been so important in getting the genotype data that we already have. And we're just indicating all the phenotypic data that is available already. So again, I've stolen an expression from George Davis Smith. He once called large birth cohorts the Hadron colliders of epidemiology and medical research. And CERN's Large Hadron Collider and the Nordic epidemiological cohort systems share some similarities. They result from public investments in science over many years. They have been made possible because of international scientific collaboration, and they are platforms that enable the pursuit of many different research questions. And I hope that they will be appreciated as such in the Nordic countries and also internationally, so that we can overcome the challenges that we see now and get the full out of these cohorts and of Moba in particular. So our task now is to show how investments in infrastructure for epidemiological science and medical and health sciences in general can make a difference to society. Thank you very much.
Thank you, Camilla. And if you stop your share right now, then I will invite Alexandra Havdahl, who is the PI of the psychiatric genetics group at Norwegian, in the Department of Mental Disorders at the Norwegian Institute of Public Health, to share your slides and start. And unmute, please.
Yes. Thank you. Thank you for the introduction. I'm looking forward to present some of the possibilities in Moba to use genetic data to understand causal influences on child neurodevelopment. First, I'll describe some different approaches to study intergenerational transmission of risk and to disentangle genetic and non-genetic influences. Then I'll give a couple of examples of ongoing Mendelian randomization studies in Moba of maternal effects on offspring neurodevelopmental problems. The first example is focused on maternal BMI, and the second example is on maternal nutritional factors. I will also outline some of our next steps and emphasize our aim to extend the collaboration with other Nordic cohorts. I'll start by giving a brief description of the background for our study examples and define neurodevelopmental traits and diagnoses. So neurodevelopmental conditions have an overall prevalence of 5 to 10% and are characterized by childhood onset, atypical or delayed development. Neurodevelopmental difficulties can range from domain-specific to pervasive across domains of communication, social interaction, motor skills, learning, and regulation of attention, activity, and impulses. Diagnostic subgroups such as autism, ADHD, language disorders, and intellectual disability are distinguished from each other based on profiles of strengths and challenges across domains. However, it's important to note that a person's profile of strengths and challenges can vary greatly, both within and across the domains, and change across the life course. Over the past decade, there's been a large increase in studies showing associations between a broad range of health behaviors and conditions in pregnancy, with offspring neurodevelopmental traits and diagnosis. Among the prenatal exposures linked to neurodevelopmental problems are maternal high pre-pregnancy BMI and low levels of nutritional factors such as vitamin D and fatty acids before and during pregnancy. Observed associations between many pregnancy exposures and offspring neurodevelopmental problems could be spurious, for example, reflecting shared family-related confounders that influence both the maternal exposure, like vitamin D levels, as well as offspring neurodevelopmental problems. One such confounding factor might be genetic risk for neurodevelopmental conditions, which in mothers may predispose to lower intake of nutritional supplements, and genetic transmission may independently influence offspring neurodevelopmental traits. Another confounding factor could be the child's nutritional status after birth. There are a range of approaches to study intergenerational transmission of risk, spanning from the classic adoption and twin studies to sibling comparison designs and extended children of twins and sibling models, where differences in relatedness can be used to estimate the role of genetic and environmental factors. There are also variance partitioning approaches using genome-wide genotype data from either family duos or trios to separate direct genetic effects from indirect or environmentally mediated genetic effects. One clear example of parental effects not explained by genetic transmission is when parental alleles that were not inherited by the child do associate with child phenotypes. When having genotype data on both parents and children, we can infer patterns of inheritance to identify the specific alleles that the child inherited and those that they did not inherit. Given that non-transmitted variants cannot be biologically expressed in the child, any association between non-transmitted variants and child phenotypes must be mediated by expression in the parental phenotypes. Decode Genetics used non-transmitted polygenic scores to estimate indirect genetic effects on a range of traits, finding evidence of associations between non-transmitted parental variants and offspring phenotypes, with the largest effects for educational attainment. This has had a great impact on the field. We're currently collaborating with Decode on non-transmitted polygenic score analysis in Moba for a range of phenotypes to better understand indirect genetic effects. I will now go on to talk about Mendelian randomization and use of trio data within that approach. The randomized controlled trial is the gold standard design to demonstrate causality, but it's difficult to use RCTs to investigate the effects of intrauterine exposures, as it's often unethical or impractical to modify them experimentally, or the long-term follow-up that's required to establish effects may be too expensive or difficult to achieve without substantial dropout. Mendelian randomization is an alternative approach to assess causality in observational studies, which employs genetic variants robustly associated with an exposure as instrumental variables to assess causal relationships between an exposure and the health-related outcome. So in RCTs, participants are randomly allocated to one treatment, such as vitamin D supplementation, or another, such as placebo. And the random allocation avoids confounding by traits or environmental factors not related to the intervention, like family socioeconomic possession. MR approaches employ the natural experiment that occurs at conception, when meiosis secures that germline genetic variation is randomly inherited from parents to children, with one allele randomly transmitted from each parent to the offspring at each position in the genome. So in MR studies, genotypes are used to stratify individuals into genetically higher or low levels of the exposure. So individuals who inherit the effect variant are essentially randomly assigned a higher dosage of this exposure than those who did not inherit the variant. As genotype is determined at conception, any associations observed between the genotype and the outcome are unlikely to be affected by reverse causation. Other qualities of genetic variants which make them useful instrumental variables are that they have little measurement error. Here is the DAG illustrating the use of a genetic instrument for maternal vitamin D levels to examine the potential causal effect on child outcomes. There are three core assumptions of MR. For MR to be valid, the alleles used to genetically instrument the exposure must be robustly associated with the exposure, independent of confounding factors, and only influence the outcome through the exposure. Assumption 1 can be tested by checking that the genetic instrument predicts the exposure robustly, limiting to SNPs with genome-wide significant association with the exposure. Assumption 2 cannot be tested directly, but checking that the genetic instrument is not associated with measured confounders can provide some evidence of the plausibility of the assumption. Assumption 3, that there's no other path between the genetic instrument and outcome other than via the exposure, can also not be tested directly, and is the one that is most likely to be violated, and is called horizontal pleiotropy. An increasing number of pleiotropy robust MR methods have been developed to assess the evidence for and to take into account potential violations of this assumption. When using MR to estimate causal effects of maternal pregnancy exposures on child outcomes, maternal genetic variants are employed as an instrument for the maternal exposure. Although an estimated causal effect could potentially reflect an intrauterine effect, there are also alternative explanations. The most important alternative explanation to exclude is that the association may simply be due to genetic transmission. This will only be a problem if offspring genetic variants affect their own outcome. For example, genetic variants related to maternal vitamin D level will be transmitted to the offspring and influence offspring vitamin D level. And if their own vitamin D level affects neurodevelopmental outcomes, then there is clear horizontal pleiotropy, and MR estimates will be biased. To date, studies have mostly addressed this by adjusting only for the offspring genotype. But as offspring genotype is determined by maternal and paternal genotype, when only the offspring genotype is adjusted for, then a spurious association is generated between paternal and maternal genotype. So then if the father's phenotype influences the offspring outcome, another source of confounding via paternal genotype is opened. Therefore, accounting for both offspring and paternal genetics is important for inferences about potential intrauterine effects. There's been limited progress in the use of MR to discover how intrauterine exposures affect child neurodevelopment. And a key reason is that large-scale family trio data have only recently become available. Genotyping in Moba, primarily done by Decode Genetics, finished recently, and quality control and imputation is still ongoing. The number of Moba family trios with currently available genotype data, as well as neurodevelopmental outcome data, range from 10 to 14,000 for age 3 and age 8 outcomes, respectively. In most of our work, we try to apply triangulation, which is the strategic use of multiple approaches with different strengths, assumptions, and weaknesses to address the same research question. In the examples I will mention, we use at least 3 approaches. Multivariable regression analysis is just running the conventional observational analysis between the maternal exposure and the offspring outcome, adjusting for known confounders. In within-family Mendelian randomization, we use maternal genetic variants as proxies for the maternal exposure in the instrumental variable analysis, while accounting for the genetics of the children and the fathers. And to further unpick whether mother-offspring associations are due to intrauterine mechanisms versus shared familial characteristics, we compare associations of maternal and maternal exposures with the offspring outcomes. In the analysis I'll present, we've used genome-wide significant SNPs from 3 GWAS to calculate genetic predisposition to BMI, vitamin D, and DHA omega-3 in Moba mothers, as well as children and fathers. These 3 maternal traits were selected as they've been considered risk factors for offspring neurodevelopmental problems and had well-powered discovery samples. So first, I'll show you some results for maternal pre-pregnancy BMI and offspring scores on the social communication questionnaire, a measure of autism-related social communication problems and repetitive behaviors. So here you see the results for 3 approaches with the offspring outcome measured at age 3. In green, you see the estimate and confidence interval from the conventional multivariable regression using measured pre-pregnancy BMI as exposure and adjusting for common confounders like the child's sex, parity, parental education, smoking, and the other trio members' BMI. The orange estimate is from the classic MR model, genetically instrumenting maternal BMI, but not adjusting for child and paternal genetic predisposition to BMI. The blue estimate is from the within-family Mendelian randomization model, additionally adjusting for child and father genetics. All of the approaches showed evidence of an effect of the mother's BMI on child's social communication score at age 3. Age 8-year estimates for classic and within-family MR were quite consistent, but with weaker evidence of an effect. Given that father's BMI would be expected to have a similar confounding structure as maternal BMI, if there really is a causal intrauterine effect of maternal BMI, then we should see a difference in the maternal compared to paternal association with offspring neurodevelopment. In contrast to for maternal BMI, there was no robust evidence of paternal BMI being associated with offspring social communication score in either of the approaches or ages. We also tested whether the child's own BMI was associated with their social communication score and found no support for this for any of the approaches. The large confidence intervals at age 3 are due to the BMI instrument explaining very little of the child's BMI at age 3. An important next step for these preliminary analysis is to expand the Moba sample size and seek replication together with our Nordic collaborators. We will also add pleiotropy robust sensitivity analysis, and we plan to use multivariable Mendelian randomization to disentangle causal effects of BMI and related traits like diabetes and hypertension, which could be driving the maternal BMI effects. We also aim to triangulate across additional approaches, such as sibling comparison design and children of twins. I'll now give the second example of MR of maternal nutritional levels on offspring score on the social communication questionnaire. The 2-sample MR analysis showed no robust evidence of any causal effect of maternal vitamin D level on offspring score on the social communication questionnaire, either at age 3 or 8. However, in this 2-sample MR design, it's not possible to separate maternal effects from the effects of offspring's own genetic propensities, given they are shared 50% with the mother. And since we did not have measured nutrient levels in Moba, we could not use 1-sample MR, where the genetic instrument, the exposure, and the outcome are all measured in the same sample. We therefore used individual level data on the maternal genetic instrument summarized into a polygenic score and adjusted for both the child's and the father's polygenic scores. So this is essentially a trio polygenic score analysis. We observed the same pattern of no evidence of association between maternal vitamin D levels and offspring social communication problems. And all the confidence intervals are quite narrow. Similarly, for DHA omega-3 levels, 2-sample MR showed no robust association with offspring score on the social communication questionnaire at 3 or 8 years. The results when adjusting for child and father DHA polygenic scores were consistent with no robust evidence of an effect. Our secondary aim was to examine whether there might be child-level causal associations between vitamin D and DHA levels and neurodevelopmental outcomes. We conducted the trio polygenic score approach using the child's own genetic instrument as exposure for the child's outcome. We found that the results were very similar as when using the maternal genetic instrument, with no robust evidence of any causal association from vitamin D or DHA to child social communication problems. We sought to replicate our findings in an independent sample and using binary diagnostic case control outcome rather than dimensional symptom scores. We used data from the most recent iPsych and Psychiatric Genomic Consortium GWAS of autism in a 2-sample MR. Again, we saw a consistent pattern of no association from nutrient levels to autism liability for either vitamin D or DHA. Due to the greater power for this case control GWAS, we were also able to run bidirectional analysis, exploring whether polygenic liability to autism might influence levels of nutrients, either through consumption or metabolism. Due to a limited number of GWAS significant SNPs for autism, instruments were selected at p less than 5 times 10 to the minus 6, leaving 39 SNPs associated with autism. We see some evidence of a causal effect of genetic liability for autism on lower vitamin D levels, indicating potentially that people with a high polygenic liability to autism may be vulnerable to low vitamin D levels. Pleiotropy robust sensitivity analysis showed that there was no evidence of heterogeneity across the autism-associated SNPs, no outlier SNPs, and no robust evidence of bias from directional horizontal pleiotropy. For the DHA outcome, we did not find any robust evidence of association with autism liability in either direction. I'll just mention some of the next steps. We want to include more nutrients and also diet patterns. And given that MR and all the other approaches have their assumptions and their weaknesses, we aim to further triangulate across additional approaches such as sibling comparison and children of twin designs. We also want to expand the Moba sample size and seek replication, especially together with our Nordic collaborators. Many cohorts and biobanks in the other Nordic countries have data that is similar or complementary to Moba data, and we hope to build on and strengthen our collaborations across the Nordic countries. In the spirit of finding similar and complementary measures across Moba and other Nordic samples, I want to highlight the types of prospective and longitudinal phenotyping in Moba of environmental exposures, developmental skills, mental and physical health, as well as linked registry data with continuous follow-up across pregnancy and birth phenotypes, primary and specialist healthcare diagnosis and treatment, prescriptions, school test scores and grades, educational attainment, income, social security, family structure, and neighborhood characteristics. The long-term longitudinal data are in particular suited to advance our field from focusing primarily on single time point outcomes to also study premorbid developmental trajectories as well as clinical progression trajectories. The factors influencing risk for incidence of a condition might be different to the factors that influence the progression after onset. The family trio data and long-term trajectory phenotypes in Moba and other Nordic cohorts will be key to advance knowledge about causes of incidence and progression of various health conditions. Before I end, I just want to mention the work done in Moba on creating an automated pipeline for extraction and cleaning of data from all Moba questionnaires and the linked registries. This allows you to search for a term, for example, ADHD, and easily get all of the relevant questionnaire items across time points, automatic cleaning and coding of scales, as well as all relevant codes from the linked registries. This work is led by Laurie Hannigan, who's also a panelist today. Thank you for your attention. I want to thank all of our collaborators and funders, and we are also very grateful to the Moba participants who make our research possible. Thank you.
Thank you, Alex. And I will, while I encourage you to submit questions, both on the bigger, everything from the bigger, larger topics that Camilla raised to the more granular aspects of Alex's presentation, we'll go ahead then and ask the panelists if you would please turn on your cameras and when I call your names to also turn on your microphones. And we will, I see Kari is here. Kari will give you the first word as usual. I can't hear you anyway.
No, I'm not surprised, how come that you cannot hear me? I didn't say anything, all right? and non-verbal communication doesn't work very well over the ether, all right? I think that the Moba in general is absolutely remarkable resource. And I agree with Camilla that it has been underestimated, the power of it has been underestimated. The possibility of studying basically everything you want to study in the context of trios is remarkable. Because, you know, genetics happens when a genetic material is moved from one generation to the next. And it is remarkable to have this large of a resource that gives us an opportunity to look at particularly the development of diseases, so development of disorders. And from my point of view, the possibility of following particularly the psychiatric health of the children in Moba provides a spectacular opportunity. The neurodevelopmental disorders like schizophrenia and autism, etc., to be able to look at it in this context is amazing. And I'm extremely impressed with the vision of the people who put this together. I mean, for those of us who are living through an era of sandbags, when no one has no one planned for more than one day at a time, to put together a resource like this that becomes more and more valuable with each year that passes. So this is basically Camilla and her people handing the next generation a remarkable instrument to work with. Knowing that Moba exists in Norway almost, not quite, but almost made me regret the fact that we left Norway 1100 years ago to settle in Iceland, but not quite there, but it almost brings me to that point. But anyway, just congratulations, for having put together this absolutely spectacular resource. That's all I can say.
Camilla, if I guess you might not want to say anything except thank you, but I will. Would you like to say anything there?
Yeah, I think the most important thing is to say thank you. but on the other hand, I mean, had I known that it would be so difficult to overcome, particularly all the judicial issues, but also the understanding of the importance of investments, I think that what we've seen in the UK, where they have invested in UK Biobank, hasn't been sort of translated into other countries, because very few countries have invested at such a large scale. And with similar kinds of investments in Norway, we would have gotten so much out of it. But we really need assistance in being able to explain why that should happen. Because so much of the focus from the authorities, from the government, from Parliament, to the extent that they have been interested in this at all, has been on sharing data, which is of course very important, but that isn't enough.
But Camilla, there is more to it than that. If you think about the UK Biobank versus the things that we are doing all over Scandinavia, and particularly when you look at the Moba, is that in Britain they have given the UK Biobank amazing freedom to work with the data. The kind of freedom that we are denied in Scandinavia today. You cannot ask people to sign an informed consent that gives us broad of a possibility of using the data. And it's interesting in the part of the world where we, in many ways, have put more emphasis on the interest of society, where we have had sort of at least social democratic societies for a long time, that somehow, when it comes to the interaction with the healthcare system, we are more focused on the right to the individual than the needs of society. I mean, completely closing our eyes to the fact that when the participants in Moba go to the healthcare system, to a hospital, with a health problem, the probability that they will be helped, that they will be getting any useful help, is solely dependent on the fact that those who came before them with the same diseases, allowed information about themselves to be used to make discoveries that are turned into method to diagnose and treat. But still, there are limitations on what you're allowed to do with the data on these people is very, very substantial. And I don't know, I think that we are somehow got lost in the woods when it comes to these issues, to put these tremendous restrictions, and it differs a little bit. My guess is that Finland is probably the most permissive of the Scandinavian countries when it comes to work on, use information generated in the delivery of healthcare for biomedical research. And my guess is that Denmark, for the moment, is probably the most restrictive, or at least it's very difficult to get permissions to use data there. But I think it's not just a lack of financial investment, it is a lack of acceptance of the fact that the healthcare system, the advancement of the healthcare system is dependent on a relatively liberal use of data that are generated in the process of delivering healthcare.
Camilla, did you want to respond?
Yeah, well, I do hope that the pandemic offers an opportunity for sort of a leap forward when it comes to this, because I mean, there has been an enormous investment in Moba. It's just that it sort of misses the last links so that we could get the most out of it. And it's been much, much easier to get funding for pretty large projects based on Moba, using Moba, than on running the infrastructure itself, which I think isn't probably unusual, but it suffers from that, so that we could get so much more out of it. But we have to be able to find the opportunities where we can communicate that and get a much stronger support for doing those parts also. And yeah, I think that it doesn't take only or we need to not only convince our own governments, but also the EU that this is worthwhile, because much of the regulatory challenges originate from the EU at the moment. Thank you.
Perhaps then we should move to Anders Berglund at Aarhus University for comments and questions.
Yeah, thank you. First of all, thank you very much, Camilla and Alex, for some really splendid presentations. I think I just want to echo what Kari said. I mean, it's just a spectacular resource you've been building up here, and it's really, I mean, great to see that you're now, I mean, not only getting fantastic results using the survey data and the register-based data, but also that you're now combining it with genetic data and can, dissect more the causal relationships that were so eloquently illustrated by the examples that Alex showed us. So congratulations with that, and thanks. I think it's an amazing resource. So yeah, and I also agree that we certainly have a long way to go yet. I mean, UK Biobank is certainly setting a fantastic example, but I do also think that we have some advantages that in the Nordic countries, obviously due to our healthcare system and our registers, and also the huge support that we have from the population at large. So I also agree with you, Camilla, that is really important to boost the investments. I know that there's been a lot of investments already, both in Denmark and obviously in Iceland, but also in Norway, Sweden, and Finland. Certainly, in recent years, I mean, it's fantastic to see this boost in investments. But still, we have a long way to go, and we could do so much more if we, boosted the investments in some way, and also were better able to share the data that we generate. And yeah, Denmark is lagging a bit behind on that currently. So, but of course, luckily we're able to share the results data in an aggregated way, so that we can collaborate across the countries. But I think it would be, of course, advantageous if we could share in a more, open fashion, or put data together to strengthen the analytical approaches. Yeah, so that was some initial comments here. I just, want to congratulate you again and thank you for the presentations and results. And I was wondering, I mean, if I should go into some more scientific questions or comments. One thing, just to clarify, I mean, obviously you've been looking into to what extent Moba is representing the Norwegian population, and whether there are any participation biases and so forth, and also what you think. When you talk, Alex, you mentioned that you wanted to expand the Moba sample. Is that possible, or are you talking about expanding the genotyping and sequencing efforts that I know is going on in DECODE?
Will you start, Alexandra, or shall I?
I can answer the question about what I meant with expanding the sample size. So what I meant was, so right now we have quality controlled and imputed genotype data on just a subsample of Moba available to us for these analysis. So the sample size is increasing.
One of the things that we could do, and we have been negligent not having done it yet, is that it really works nicely to impute rare variants from sequences in Iceland into Norwegians. Because in spite of these 1100 years, we still share a lot, all right?
Yeah, that sounds fantastic. So how low can you go in terms of minor allele frequency?
We can go pretty low. I think we could probably not go quite as low as we can in Iceland, but we can be approaching that.
That's cool.
Right. So we are still left with the burden of our heritage. We are still left with the burden of being related to the Norwegians. So it's about time that we can take advantage of it. Get paid.
Go back and respond to the first part of your question, Anders, about the selection bias. There is some selection bias, but the thing is we have pretty good control over it because we have so much data on the population at whole. And the Moba participant parents are slightly better educated, slightly older. Some a higher proportion are married and cohabiting, and so we know about all these biases and can assess them in each study. And so far there haven't been any major problems with the biases. But the main challenge is that the population of immigrants in Norway is not represented at all. So basically, that's a fairly high proportion of people in Norway, and that means we need other sources of information if we want to study them also. Basically, or mostly registry data can be used.
Perhaps we should move and give Laurie Hannigan a chance to make a comment or a question, and then we'll go to Red Stefansson, and then back to more general discussion.
Yeah, sure, thanks. So I had a few things to pull out from both of the presentations that I think reflect some of the challenges of maximizing the utility of Moba and other Nordic cohorts and data sources that, and I can maybe ask for your thoughts on. One was what you've just been discussing about, Camilla and Anesh, about the representativeness, and I was thinking in particular of examples from UK Biobank, which has been referenced a couple of times already. You know, for example, recent work showing in genetic analyses that finding autosomal variants associated with sex in UK Biobank is giving an indication of, you know, the breadth and spread of sex-related participation biases in UK Biobank. And I think when we think about representativeness in the likes of Moba, especially as we get into these sort of finer grained genetic analyses, it's all the more important to consider those aspects that you've both mentioned already. The other point I was going to mention from Camilla's talk that you brought up was about this question of impact and making an impact with the work from Moba, and you referenced the Large Hadron Collider sort of analogy, and I've never seen the impact statement on a grant from a physicist from the LHC, but I'm guessing it's easier for them to sort of recourse to, you know, the general wonderment and magic of the origins of the universe in terms of their impact statements, whereas we in sort of health science, I guess, have more of an obligation to find these direct, applicable, kind of clinically informative impacts of our work. And that's clearly, I think, a challenge, an ongoing challenge. And then a couple of points from Alex's talk. So you mentioned about triangulation and this idea that I don't know who to attribute to the quote to, but I've heard it said that a discipline is just a group of scientists who kind of collectively agree to not question the same assumptions. And I think we have to be careful that with the similarities across the Nordic region, with all these great data sources, we're not sort of a collective of people who kind of ignore the assumptions that are common with analyses of these data. And finally, the other thing, Alex, in your talk that you mentioned that you showed a slide with me on it, so it shows that it's one of my interests, but around open science and the options for reproducibility and code sharing, especially in the context of the inability of data sharing, which has been referenced already in discussion. So the fact that we have restrictions on being able to share data across sources in these from these data sources, I think makes it all the more important that we find ways to build reproducibility and build kind of a collectivist approach to our analyses in other ways. So I guess my question then to both of you is are there any of these sort of challenges or any others? So across representativeness, impact, triangulation, or openness and reproducibility, or any others that you think researchers using Moba and other Nordic kind of registry and data cohorts should be particularly focused on in order to maximize the utility of these sources?
I guess I can start. Maybe Camilla wants to comment. Very big question. I think that the focus on triangulation is very important to me, and I'm sure that you get blind by your own assumptions, but focusing on combining very different approaches, or approaches that have different sources of bias and different assumptions to each other, then you'll get a more coherent picture. So I think that's very important going forward.
I'm not sure I can add that much more to this. I think the questions or the issues you raise are extremely important, and we have to consider them as we go in all projects, and always look for opportunities to reproduce and to triangulate and to challenge our assumptions. But again, to some extent, this goes back to the broader issues. Why don't we invest? And I mean not only invest money, but invest in sort of preparing society for being able to run such studies and to reproduce them, and to do that efficiently without all the hassle that we all have to go through, and all the uncertainty about whether we will be permitted to do things. I'm very sort of influenced at the moment by what we've experienced during the pandemic, where suddenly we've been permitted to do a lot of things that we have to wait for years, and sometimes without knowing when we will get the results. So now we've been able to do it, and I think we should use that as a demonstration of how efficient and good that is, and it doesn't mean that confidentiality and all the data security, etc., has to suffer. There are solutions to these things. If it's important enough, it will be solved. And it's not as if those solutions that we have today, when it's so cumbersome, are better than the ones that we could have if we decided that this is so important to society, both to have the long-term kind of science that doesn't necessarily have an impact immediately, but that is there as a resource when we have made new discoveries. We see that it does eventually end up in some kind of application, some fast, some very slow, but also the more applicable science, where high quality is needed equally much as when we do science that is not applicable. And I think by using the pandemic to illustrate these issues, and by using it to show politicians and authorities that will give us a better society, and that will give us economic opportunities, we should be able to make these kinds of resources into something that everyone just has to have. And that becomes a much more natural part of society, where researchers can do the research, but others can build the infrastructures based on what's needed. So that doesn't answer your question directly, but I think that is the main reason why we don't replicate, or why it's so hard to do it, because data are so hard to get, and collaborations can be so difficult to establish.
You see, there are two things about this that I'm a bit concerned about. This call for standardized approach to science. Like everyone is doing the same thing, and then it's so much easier to get replication of work that has been done. But I think it is, and when you look at the journals these days, everyone is pretty much doing the same thing. There's very little, or I find very little creativity in the way in which people are approaching their study of human diversity in general. So at the same time as I think it is very important to be able to reproduce what you have done, it's extraordinarily important that you as a scientist try to do something that is truly new. Am I being still another time condescending and arrogant? But I think it is very important not to impose vanilla science on everything. We have to do something a little bit new. That's number one. Number two, I think that this emphasis, this emphasis on privacy, this emphasis on the protection of privacy in healthcare data is a little bit twisted, because it doesn't matter where else in society you go, data on individuals are available all over the place for all kinds of uses. And it is rarely that it is complained about. People have access to financial information through their credit card companies and stuff like that. But when it comes to data that you're going to use just to try to discover something new about nature of disease, then it all of a sudden becomes very dangerous. And then you take a step back and you ask the question, when is the last time that you know of that healthcare data gathered for the purpose of doing biomedical research were abused or used to hurt people? And the examples are extraordinarily few and extraordinarily far between. And I think that we have to be somewhat careful in not ourselves placing too much emphasis on the privacy stuff. Try to convince the world that we can protect data, personal data on healthcare, in such a way that no one will ever be able to break into them, because the fact of the matter is that you can never completely protect data, all right? It's very difficult to do so. And particularly currently, when everyone is using, cloud systems to store and mine data, it becomes basically impossible. We aren't, what I'm trying to tell you is that you aren't doing anything indecent. You're trying to make discoveries. I don't think you're particularly good at it, but you're at least trying to.
Maybe we should let Rhett go, but I do see there are some specific questions. So Rhett, would you like to jump in?
Yes, I would like to ask Alexandra a couple of questions. So, and this has to do with vitamin D. And Norway is kind of interesting country with respect to vitamin D, stretching almost from Central Europe up to the North Pole. So you mentioned that you had vitamin D levels in mothers, and are those data from basically before pregnancy, or is that during pregnancy? And the reason why I'm asking is that the two phenotypes that you are focusing on, among others, are ADHD and autism. And ADHD associates with basically lower educational attainment. And lower educational attainment, if I'm correct, and please correct me, has been associated with higher vitamin D levels. So what I'm asking about basically is the data on vitamin D. How are they collected for this study? And do you take into account basically year of birth, etc. also, because that can be affected by the sun, etc.?
Yeah, great question that I should clarify. So in this study, we only had the genetic predictor of vitamin D levels. So it was used in a 2-sample Mendelian randomization, where the one sample was the Manusaki GWAS of vitamin D levels. And then the outcome GWAS was in Moba. But we really would want to also have measured vitamin D levels in the mothers, so that we could compare and especially check that the genetic instrument for vitamin D actually predicts maternal vitamin D level in pregnancy. We weren't able to test that in Moba now, because the sample with the vitamin D measurements was just too small, and we haven't been able to link it to our data source yet, but we hope to do that. We checked it in Alsbach, another birth cohort, and check that the genetic instrument does predict vitamin D levels in mothers during pregnancy. The link to educational attainment is a very good point. One thing we hope to do when taking this further is to do a multivariable Mendelian randomization where we take into account both educational attainment and vitamin D levels, just to see whether that could be driving some of the association.
Yeah, what I was maybe thinking about a little bit is that if you have some estimates in Norway, whether there is difference in southern part versus northern part, is that something you should kind of correct for, or should you analyze separately the samples from northern Norway versus southern Norway to see if there is more difference?
Right, so we could see if it, so when we use the genetically predicted vitamin D levels to see whether the exposure to sun may influence the association. One thing that we thought was interesting, we've looked at whether there are any systematic differences in the diagnosis of, for example, autism in the various counties in Norway, and there is no systematic increase. There's no increase in the rate in the north, which is consistent with the results from those analyses. But if we use the measured vitamin D levels as opposed to the genetic predictors, we would absolutely need to take into account the geography and all of the factors that play into the season, everything that play into the measurement of vitamin D levels.
Thank you.
No, I'm not completely updated on this, but last time I checked, which may be close to, well, 8 years ago or so, the differences in vitamin D levels in Norway were not dependent on geography. They were dependent on educational level, which again was associated with supplements and diet.
Mm-hmm. The consumption of cod liver oil.
We have a question from Søren Dalsgaard. He asks, he notes, Moba mainly have data on symptoms for the large sample and fewer participants with diagnoses. I'm sorry, data on symptoms for the large sample and fewer participants with diagnoses. How similar are participants with a high symptom score for example, ADHD to those with a diagnosis of ADHD? Have high scoring people in Moba been used as proxies in GWAS? Would this be possible?
So I think, yeah, measures of dimensional traits or symptoms is definitely different than a clinical diagnosis. For example, the clinical diagnosis will take into account many other factors than here. It would be maternal reported symptoms of autism, whereas a clinical diagnosis also takes into account functional impairment and the functioning in many other domains. And one thing that is unique about Moba is that you can use both, that we have diagnosis from the registries in the full Moba sample, which for many purposes is good to be able to compare whether there are differences between for associations when you use like high scorers versus people with a diagnosis. Although in Mendelian randomization, we do not have the power to look at the diagnostic groups for most things within Moba. but in meta-analysis, that's possible.
I could
Anders,
probably
you have a comment?
Yeah, I could probably add, I'm sure also, Alex, that you're aware of that the genetic correlation between specifically ADHD, diagnosed ADHD, and symptoms of ADHD in the general population, the genetic correlation is extremely high. It's close to 1, and we did include it in our first ISAC PTC GWAS of ADHD. Did some replication, and it's also the ambition to include symptom-based scores in the quantitative GWAS in the upcoming ADHD GWAS from the PTC. And I think actually that Moba is contributing to that part as well.
Yeah.
And so yeah, just to add that perspective as well. But clearly it's different when it comes to social communication and correlation with autism diagnosis. The correlation is far from as high.
Paul Njølstad asks to Camilla, are there plans to perform follow-up studies either by questionnaire or the more valuable physical investigations and samples, and would it be possible to follow the Hunt example of 10-year follow-ups?
We are following up with questionnaires much more frequently than every 10th year. So we do that both based on specific issues, for example, the COVID pandemic, but also on a regular basis. So that's being done. Regarding the collection of biological specimens, that we haven't been able to fund that for the whole cohort. So that's been funded based on specific projects with subsamples. But we would very much want to do it. But we don't have any concrete plans for it.
Rune Bøen asks, how is the volunteer bias distributed across Norway? For example, is it a stronger bias in the northern part compared to the southern part?
Well, that's relatively easy to answer because it's not very different in the different parts. But for northern Norway, they didn't participate with the births in Tromsø because Tromsø University Hospital and the university didn't want to participate. So that was one of the 2 out of 52 clinics that did not participate in Moba. So that means that the participation is much lower in among those people who gave birth in Tromsø.
Any other comments or questions from the panelists? If not, oh yes, Anders.
I have a question to Alex. I think it's a tremendous resource that you have all these trios, and also from a genetic perspective, I think it's really interesting in many ways. But I was thinking, I wanted to ask you whether you were looking into parent of origin effects to see if there are imprinting like effects on the associations you see or that others have observed, and whether you can see that kind of parent of origin effects in your cohort.
I know there are several efforts going on to examine parent of origin effects. So in my group, we haven't focused on that yet, but there are several ongoing efforts that will be presented this year at the World Psychiatric Conference. So I will be sure to let you know about an update. But that is in the early days.
Thanks.
Any closing thoughts from Camilla or Alex if we as we head toward the end here?
No, just thank you very much for inviting us, and I was so glad it happened at this very moment when it was possible to focus again on these issues and to be inspired to pursue the further development of Moba and further use of Moba, particularly in collaboration with other Nordic countries that have similar resources or complementary resources. So that would be wonderful to do. Thank you.
Thank you very much for having me.
Thanks to everyone. I will say also thank you to the panelists, and I will leave you with, I think you might see the advertisement for our conference. Please submit your abstracts, your best science, and to both the workshop and the main meeting, and register.
Thanks.
Thank you.